Kia ora koutou, ko Steve Pawson toko ingoa, e mahi ana au ki te kura ngāhere, ki te whare wānanga o Waitaha. Kia ora, Steve Pawson here from the School of Forestry at the University of Canterbury. Many of you probably know me from my past role at Scion, leading the entomology team. Now I'm here at the University of Canterbury working in forest health and biosecurity. So today I'm going to be talking about some of the pests of dryland eucalyptus. So the New Zealand Dryland Forest Initiative was formalised in 2008, although it started a little bit earlier than that. And the focus of the program is to develop a naturally durable source of timber to replace CCA-treated pine. So the program itself is investigating a number of species, but there's 5 in particular that they're taking forward in their breeding program. So what we know about eucalyptus is that, yeah, it's got this naturally durable timber, but also insects are somewhat attracted to eucalypts. And we've been receiving quite a few of them from our friends in Australia, which is the, shall we say, centre of diversity for eucalyptus, but it's also the centre of diversity for eucalyptus pests. And we're just downwind, so not only do we get the smoke from their bushfires, but we also occasionally accumulate insects from their forests that blow over or hitchhike with us on international trade when we're moving goods between Australia and New Zealand. So we've been receiving insects from Australia for quite some time now. Some of the earliest invasive forest insects came from Australia and fed on eucalypts, things like Voracantha, semipunctata that arrived as early as the 1860s. And we've been steadily accumulating the odd thing ever since. So the situation today is that there's 2 really key defoliating species that are a real big problem for trying to expand the eucalyptus growing industry to a larger variety of species, but also to protect the fairly sizable Eucalyptus nitens plantations. So these 2 species are both pyropsene beetles, and one has been here for over 100 years, Pyropsis charybdis, and Tony Withers has spent most of her career looking at biocontrol options for Pyropsis charybdis, otherwise known as the Eucalyptus tortoise beetle. But more recently, another significant defoliator that's possibly going to be worse than Pyropsis charybdis arrived in 2016, and this people probably know as the Eucalyptus variegated beetle. It was known by the scientific name Pyropsis verrucolis, but when looking into that particular species in more detail, they have recently found through molecular studies that it's actually a complex of different species, and the one that we have is a thing called Pyropsis cloalia. So a key part of what Leslie's trying to do for her PhD is to examine whether or not some of these eucalyptus species that form part of the Drylands Initiative exhibit what we call resistance or tolerance. So there's quite a big difference between these 2 things. So resistance is where the herbivore doesn't really want to eat the plant. So it doesn't taste good. So either there's nasty compounds in the leaves, or there's some sort of tough fibrous nature to the leaves that the insect doesn't want to feed on that particular plant. The alternative option is that the plant has developed strategies to cope with the feeding. So it's a more stable coevolution than resistance. Resistance is a bit of an arms race. One develops a defense, the other one tries to overcome the defense, then it needs a new defense. But in terms of tolerance, the plant has learned to live with it. It doesn't mind having having a bit of a nibble on its leaves, and it continues to be productive, put on height, put on DBH. And so those are the kind of things that we're looking for when we're trying to make breeding decisions around protecting these eucalyptus species from herbivores. So as we go on, we'll be talking about resistance and tolerance. So I mentioned earlier that there's 5 key species that the Drylands Initiative is interested in as part of their main breeding program. And these fall into 2 subgenera of the eucalyptus. And you may have heard of these names. There's the Symphomertus and the Monocalyptus. Now most of these are Symphomertus, and unfortunately what that means is the insects think they're like chocolate. So insects in general prefer the Symphomertus eucalyptus than they do the Monocalypts. So the colours that I've got on the presentation here are quite important. So the red ones that I show you on the next slide are the Symphomerts, and the blue ones are the Monocalypts. So let's take a look at some of the previous work that's been done by Hui-Min Lee, who finished her PhD a few years ago, and she looked for evidence of resistance. And so she examined a range of species using a fairly coarse measure of how much the herbivores were feeding on the trees. So damage levels A, B, C, and D. So D, the darker the colour, the more damage that these have. And what you can see is that by and large, the more heavily damaged species were the Symphomertus. The one that's probably the exception there is Eucalyptus cladocalyx. What we don't know is whether this resistance is heritable, so can it be passed on genetically as part of a breeding program? That would be a latter topic to be looked at. But what we can see is that there's 3 species, cladocalyx, macrorhyncha and globoidea, that seem to be the ones that are most resistant to the browse of these 2 paropsines. So Leslie is taking the work of Huimin further, and she's using an index of defoliation called the Crown Damage Index. So higher values of the Crown Damage Index mean greater defoliation. And one of the really pleasing things that we can see in the very early stages of Leslie's PhD is that she's seeing the same results that Huimin did. So Huimin was working in the North Island largely, Hawke's Bay, and these results are from the South Island, from the Marlborough region. And we're seeing the same 3 species, the macrorhyncha, the cladocalyx and the globoidea, having lower levels of defoliation than all of the other Symphomerts. Cladocalyx is still that standout oddball that seems to have low defoliation, despite being one of these Symphomertus species. So there's quite a large range of damage between species, which suggests that there is some opportunities to select for species that are more resistant to the browse by these particular paropsines. But I guess the big thing that we want to know is how is this affecting growth of these Eucalyptus species? So this is very preliminary data for Leslie. So she's in the first year of her PhD and she's taken measures of defoliation, which I showed you on the previous graph. And this is the first measure that she has of the growth of these trees, so the DBH of these trees at that point in time. Leslie's subsequently going to go back and she's going to resample these plots, again looking at defoliation, but again measuring the DBH. So we can see what the growth increment will be over the course of 1 year relative to the defoliation that they suffered. And what we can see at these particular sites, Dillon and Leeseman, so the left and the right columns in each little pair is Dillon and Leeseman. They were established at different times, so we would expect the DBHs between the 2 sites to be quite different. But what we can see is that those species that were less defoliated, we're seeing on average a greater DBH. Those are the horizontal lines that I've put, sort of just a rough average in there. And there are these outliers that are much greater than the average. So what we need to do now is try and figure out, is that an environmental effect that's causing that? Or is that something about the genetics of those species that we can work with that might be repeatable in other locations? So are there some species that are more resistant? And over the years, as Leslie collects more information about growth as it occurs with the defoliation, we'll get an idea if that is expressing tolerance as well. So one of the really critical things that is going to allow us to expand on this work is to find rapid ways to assess defoliation of eucalyptus on a large scale, so a whole site basis. So to date, we've been reliant on things like pest counting, tree grading, which is Huimin's work, and the Crown Damage Index that Leslie's been using, but these are very time consuming. They require an individual person to go and assess every particular tree, potentially cut shoots off those trees, bring them down and assess them individually. You can't do that at scale, and so therefore we can't support big breeding decisions that need to be made at scale in terms of assessing a whole pile of different families and clones across multiple sites to sort of examine if there's side effects. So the way forward that we want to go is we want to go aerial. So let's get some eyes in the sky and use some imagery, be it RGB imagery or LiDAR. Leslie's going to be working as one of the projects in her PhD is to evaluate, can we come up with a method to evaluate the level of defoliation of these eucalyptus in a repeatable manner that we can then correlate with the levels of defoliation that Leslie is observing on the ground. So the idea is to get geospatial data or remote sensing data that we can then correlate with the actual observations that we make on the ground of the levels of defoliation. And that'll be a really useful tool going forward that can not only be used to make, shall we say, decisions for breeding, but also could be for management decisions in eucalyptus plantations, i.e. have things got out of hand and do we need to implement a spray perhaps to knock the population back? So talking about future plans, we actually have 2 PhDs on offer at the moment. So if you know any bright young students that want to come and work with me and Tony Withers, one of them is going to be on the Eucalyptus variegated beetle, so this Paropsisterna cloalia, and we're going to be looking at how some of the generalist predators, like the Clearbora ladybird beetle in the image there, might help control the pyropsene beetles. So how might we be able to make life better for the ladybird such that it can have a good go at these pyropsenes? And one of the things that we're quite interested in looking at is having mixed stands where we have a number of different tree species in there that get cut out during the thinning process, but these species will help the ladybird beetle thrive and hopefully exert more control, thus resulting in better outcomes for the main crop species of the eucalyptus. So as I mentioned earlier, the pyropsene beetles are the key defoliators of eucalyptus in New Zealand, and Tony, Withers at Sign and others have been working a lot on parasitoids of these species for other reasons other than the dryland eucalyptus project. Now what they've found is that one of those particular parasitoids, Anogra, is actually pretty good at parasitizing the new beetle Parasitoids cloelia, the Eucalyptus variegated beetle. Almost 50% of the eggs in one study in the North Island where they were looking at this were parasitized by Anogra. And that's a great result, which means that we can look forward to hopefully having some good control by this species of the variegated beetle. Now the unfortunate thing is the other parasitoid, Neopolycystis, it also attacks the new Parasitoids cloelia, but unfortunately it doesn't result in viable offspring. So it's a bit of a double whammy in that we're not actually controlling the cloelia, but also they're kind of wasted effort by these little female parasitoids that could otherwise be stinging some Paropsis charybdis eggs and dealing with the other species. So many of you are probably aware that the EPA recently approved the release of Aedia daenerys. Now this is a new parasitoid, not currently present in New Zealand, and Tony and others at Sign have been working very hard for many years to get all the host specificity data, and it was recently approved. So over last Christmas they were working hard trying to breed up many individuals from a cohort that they brought over from Tasmania, but unfortunately they couldn't release them into New Zealand outside of Sign's containment facility because it was found after they were all bred up that they were contaminated by Microsporidia. So big disappointment for the team, but they're going to be trying again this coming December. So Aedia daenerys is another parasitoid of Paropsis charybdis. So that's great news for controlling Paropsis charybdis, but it doesn't attack cloelia. Now, as part of the release process or part of the research that's gone into Aedia daenerys, what we found, or what an international team that Tony's been part of has found, is that actually the Aedia daenerys is part of a species complex. And there's actually 3 or 4 species in there, and one of them, Aedia andlecii, does go after Parasitoids cloelia. However, to get permission to release that in New Zealand, we have to go through the whole host specificity testing. Now that's quite a large piece of work, 2 or 3 years worth of work, but I think it's probably worthwhile that the industry starts thinking about that if they want to have an expanded eucalyptus industry, because it's likely, or what we're seeing so far in the field, is that Parasitoids cloelia is going to be as bad as charybdis or potentially worse. So currently we don't have any specific focus parasitoids on that species, so it'll be good to think about Aedia andlecii for the future. So I guess the key message is that eucalyptus is like chocolate. Everyone kind of likes it, but they like some flavours more than others. And that's what we're finding with these pyropsene beetles. None of the species are 100% resistant to browsing by these species, but definitely some species seem to be more resistant than others at this point in time. However, because we're not seeing really high levels of resistance by any of these species, we really want to understand this tolerance. So how tolerant are they of a little browse such that they can continue being productive and putting on the DBH and the height? So that'll be a key focus of us going forward, is understanding tolerance. And for us to make the biggest leap forward that we can in understanding tolerance means that we have to be able to assess defoliation at a really big scale. We need to be able to do it at the trial scale, so we can be looking at multiple species, multiple families and clones all at the one time. So that'll allow us to understand the most around how tolerance is happening and what site effects are. The other thing that goes alongside breeding for resistance and tolerance obviously is the biological control. So how can we exert top-down control to reduce populations? We know we have some parasitoids that are there already. We know we have some generalist predators like the Clearbora ladybird beetle. So how can we maximize those particular parasitoids and predators to control populations? And also, do we need to start thinking about new parasitoids, as I said, Edia and Lecii, and ensuring, of course, that we get Edia daenerys here and well established in New Zealand. So that's it from me, a little bit of a roundup of stuff that's been happening in the past, some of the stuff that's happening now, and where we plan on going in the future. So if you have any questions, feel free to ask them during question time or drop me a line at the email on the screen.